Welcome to another Biochemistry MCQ series. My name is Dr. Raymond Ibe. So we'll start with this question. Which of the following best describe waxes? They are hydrophilic, carbohydrates that are brittle. No, they are not carbohydrates. They are lipophilic, organic molecules that are malleable. Okay? They are inorganic salt. No, of course, they are not inorganic salt. They are protein, they are not protein. So the best option to this question is option C. Option C. Option B. Option B. They are lipophilic, and they are usually made up of long chain fatty acids and long chain alcohols. Note, they are made up of what? Long chain fatty acids and long chain alcohols. Now, they usually have a melting point of above 40. A melting point of above 40 degrees Celsius because of they form compact. Of course, you've seen like candle wax, wax forms compact. So they usually have a higher melting temperature. So the answer to that question is they are lipophilic organic compounds that are malleable. So the high melting point gives it its high viscosity. So the viscosity of the wax also makes it high. They are usually produced by plant and animals. Remember I said they are long chain fatty acids and long chain alcohol. So you have something like this. Long chain fatty acid, usually from maybe 10. This is a fatty acid, OH. The long chain alcohols, OH, C, 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 C. So you see something like myristoy and palmitic acid joined together. So it will be myristyl palmitate. To be myristyl palmitate. So it will be the carboxylic end and the hydroxylic end will react to form esters. So waxes are esters of long chain fatty acids and long chain alcohols. So which of the following is the most well-known wax? The answer to that question is the B wax, the well-known wax. The most abundant sterols found in animal is cholesterol. Cholesterol is the most abundant sterol that is found in animal. Ergosterol is usually found in fungi. And that is what some of the antifungi cream inhibits. Ergosterol biosynthesis, like those cotrimazole inhibit ergosterol biosynthesis. Cytosterol is a phytosterol found in plants. Stigmasterol is also a sterol found in plants. So the answer to that question is cholesterol. Which compound acts as a precursor for steroid hormones and fat-soluble vitamins? The answer to that question is cholesterol. Cholesterol is what is used to produce the steroid hormones, the testosterone in man, in male, the estradiol, estrogen, estrone found in female, even progesterone, they are all produced from cholesterol. Sterols are naturally present in the following except They're usually found in animals, they're found in plants, they're found in fungi. Remember I told you the fungi own is a good sterol, found in the plant, found in the fungal membrane. We have the plant which is phytosterol, campysterol, stigmasterol, cystosterol that are found in plants. In animal we have the cholesterol. Sterols that are derived from fungi are known as what? Ergosterol. Ergosterol is derived from fungi. Ergosterol is for fungi. Phytosterol is for plants. Cholesterol is an animal sterol, while zoosterols are actually used to define sterols that are of animal origin. Sterols that are of animal origin. Sterols originating from animals are referred to as what? Zoosterol, like I said in the previous slide. Phytosterol, mycosterols is fungi of fungal origin. Glycosterols is sterols that are attached to a carbohydrate molecule. So the answer to that question is zoosterol. Plant sterols are essential component of all cell membrane. This question, of course, is eukaryotic organism, but there is a somewhat, comma, or let me say somewhat, part of this question, because if we say plant sterols, the plant sterols are not found in animal cell membrane. They don't found in animal cell membrane. And animals are actually eukaryotic organisms. They're eukaryotic organisms. Phytosterols are synthesized through dash pathway. Through dash pathway. The phytosterols, glycolytic pathway, no. Glycolytic pathway is for breakdown of glucose. Pentose phosphate pathway is to synthesize 5 carbon sugars and NADPH. Krebs cycle is where most of the food molecules are actually, it's a central hub that connects carbohydrates, lipids, and protein metabolism. So the answer is mevalonate. The answer is mevalonate. Mevalonic acid. The answer to that question is mevalonate, which is actually the committed step in the synthesis of cholesterol or other sterols. Now, that pathway is catalyzed by an enzyme called HMG-CoA reductase. HMG-CoA reductase converts HMG-CoA. HMG-CoA is a beta-hydroxy beta-methyl glutarate coenzyme A. So the enzyme that acts on it is HMG-CoA reductase. The enzyme converts HMG-CoA to mevalonate, and that is a committed step in the synthesis of cholesterol or phytosterols. Now, for human, if we want to inhibit cholesterol synthesis for people that have hypercholesterolemia, we use the statin drug, the simvastatin drugs. We call them generally the statin drugs. So they are used to inhibit cholesterol biosynthesis. So do not forget to subscribe and click the notification bell for more videos on biochemistry MCQs.